Thank you. Firstly, thank you, Dr. Meekins, for this amazing platform, which I think will be so helpful for early career researchers like me. Today, I'm going to talk about, I am a postdoc at Harvard Med. Today, I'm going to talk about some of the research that I did as a graduate student in Dr. Smita Patel's lab at Rutgers. The topic of my talk today is mechanism and regulation of mitochondrial DNA transcription. As you know, the central dogma dictates that genetic information that is coded in the DNA gets converted to RNA and protein through the processes of transcription and translation. I specifically study the process of making RNA, which is transcription. And whenever you think of transcription, you obviously think of the nucleus, where your the information in your genome is coded into mRNA that makes proteins. However, let me remind you that the genetic material in your nucleus is not the only genetic material that you have. We have a whole new, we have a separate set of genetic information that is coded in a completely separate genome, which is the mitochondrial genome. And mitochondria has its own genome. It is, as you know, an organelle in the cell, and it has its own genome. Tracing the origins of mitochondria, I think what the general consensus is that around 2.7 billion years ago, there was an endosymbiotic event where prokaryotic host cell, an ancient bacteria, engulfed another smaller bacteria, an alpha proteobacterium-like ancestor, and this gave rise to a primitive eukaryotic cell, which had two sets of genome, its own nuclear genome, and then the mitochondrial genome that it acquired from the smaller bacteria. Thus, this proto-eukaryotic cell slowly evolved into, I'm going to get, is there a, this proto-eukaryotic cell eventually became the modern eukaryotic cells, and it still maintains its own mitochondrial genome and a separate nuclear genome. As you know, mitochondria is crucial in health and disease, and here, and the main reason is mitochondria is the powerhouse of the cell in that it makes energy currency in the form of ATP. And this ATP is used by high energy consuming organs in your body, like the heart, eyes, liver, kidneys, to perform a lot of biochemical reactions. And failure or decrease in the amount of energy that we supply to these organs can cause cardiomyopathies to diabetes to cancer, etc. Thus, it is crucial that we understand the basic processes of maintenance and expression of the mitochondrial genome that can help us understand the malfunctions of this organelle. So, like I said, mitochondria has its own RNA polymerase, has its own genome. So it's a separate organelle, it's a world of its own, other than the nucleus. It has a circular genome, which has its own polymerase to transcribe this genome. So this is a very autonomous system that exists in your cell. Think of it as a small world. Curiously, mitochondrial RNA polymerase is homologous to the T7 bacteriophage RNA polymerase. So it has the mitochondria in your body, the polymerase actually resembles a viral polymerase. So as you can see from this figure, both the yeast and the human mitochondrial polymerase, they are structurally highly similar to the single subunit T7 bacteriophage polymerase. And the classic features of the polymerase, like the palm domain, the fingers domain, the thumb domain, that are used for promoter recognition and melting, are all conserved between these three polymerases. So the main structural similarities are towards this C-terminal part. And as you move towards the N-terminal part, you see more and more dissimilarities that come up. However, in spite of being very similar to the single subunit T7 RNA polymerase, mitochondrial polymerase is unique in that it requires the help of additional factors or proteins to carry out transcription. So the bacteriophage T7 polymerase is pretty independent. It does not require any accessory factor for transcription. It can do everything on its own. In Yeast mitochondrial system, we have one factor, which we call MTF1, help the main polymerase RPO41 to transcribe the DNA. And in the human system, we have two factors, TFB2M and TFAM, that, and these are the factors that help the main polymerase, RMT, to transcribe RNA. So I think the big picture in the field, the big question in the field is that why, in spite of being so similar to bacteriophage, why does it require factors to initiate transcription? So here is a small animation of how this whole system works. So we have the first protein, which is TFAM. The first factor comes and binds to the DNA, and this now characteristically bends the DNA, and this is kind of prepping the DNA for transcription. Next, the DNA is bent, and this recruits the polymerase, which is recruited to this complex by a very specific interaction with the tether helix, which is shown in green here. So now there's a DNA, there's a polymerase, and one factor. Once this structure is formed, the second and the final factor, TFP2M, enters the complex. Now, TFP2M, the main function is to reposition some elements of this polymerase that can now help melt the DNA. Thus, these three proteins with the DNA forms what we call the human mitochondrial initiation complex. Now, it is pretty tricky to genetically manipulate mitochondria. So what we do in our lab is we in vitro study the proteins that are involved in mitochondrial transcription. So we basically clone genes in E. coli. We use the prokaryotic protein expression system, or the bacteria, to express these proteins. And then we study these proteins in vitro using biochemical and biophysical methods. So for those of you who don't think of transcription every day, I think this schematic is a very simplistic, will give you a simplistic idea as to how transcription happens. So when we think of transcription, in the first step, we have the promoter binding step, where the enzyme, which is your polymerase, comes and binds to the promoter DNA to form the enzyme DNA complex. In our lab, we can study this process using the biophysical technique of fluorescence and isotropy. In the next step, this DNA needs to be wedged open so that the polymerase can now read the template strand or the message in the DNA. So the next step is melting of the DNA strand to form the open complex. And in our lab, we can monitor it using various fluorescence methods like 2AP fluorescence. The next step is when the initiating NTPs, shown here with these blue dots, come and bind to the polymerase, and the subsequent formation of the phosphodiester bond between the two nucleotides in this RNA that will form the tumor RNA, and this is the first chemistry of the RNA, the first reaction that joined the two nucleotides. So when transcription initiation happens, this is how the steps are brought about. And this is true for any polymerase, any transcription system that you're studying. And the last two processes in our lab are studied by various fluorescent and gel electrophoretic methods. So let me give you a brief structural idea about the homologous factors, TFB2M, which is in human, and MTF, which is in yeast. So as I said, these are mitochondrial transcription factors. These are, curiously, these factors are similar to methyltransferase enzymes that transfer methyl groups to various proteins. So we think these might have evolved additional functions of transcription during evolution. So the basic structure consists of a very conserved N-terminal domain, which has the S-adenosylmethionine or SAM methyltransferase domain. It has 7 beta sheets surrounded by alpha helices. And then there's a smaller C-terminal domain in both, which is entirely composed of alpha helices. And curiously, the C-terminal domain also has a kind of an extension, which is around 15 to 20 amino acid long C-tail, which is shown here with dotted lines, indicating that the structure is not resolved with X-ray crystallography. Now, as you may know, that transcription factors are notorious for having very flexible regions like these. They are flexible and very, harmless looking parts of the protein. But once they come in contact with the polymerase and or DNA and or other factors, they can assume very specific structures and can carry out very specific functions in transcription. So we were interested in knowing the structure of this C-terminal tail in mitochondrial initiation in both yeast and humans. So before I started this work, this is what was known about the C-tail. It was not resolved in crystal structure. The main protein, the main body of MTF cross-linked to the unbound non-template strand, and the C-tail cross-linked to the -3, -4 template strand. And this reminds you of the sigma 3.2 region of the E. coli polymerase. So sigma factor is one of the most studied transcription factors in the bacterial system, of course, and the 3.2 region exactly cross-links with the -3, -4 region, which is very similar to the C-tail. So from here we were like, so is this part homologous to the sigma 3.2 region? That was our question. And then also in 2015, a paper came out that said the C-tail was involved in promoter melting. So in 2017, we had the structure of the human mitochondrial initiation complex. And as you can see, TFP2M, the factor here is shown in pink, and the polymerase itself is shown in white. You can see the thumb domain of the polymerase, and I have drawn the C-tail with this purple-blue color. And as you can see, these residues have very specific interactions with the polymerase. And I have also modeled in the 3-mer RNA structure, and you can see this is very close to the RNA, and this is in the absolutely in the active site of the initiation complex, which shows that it is right there in the active site. And this was very interesting to us, which indicated to us that it might have specific functions in initiation. So what we did is we took both the yeast and the human homologous transcription factors, and we made specific deletions in the C-tail to make C-tail deletion mutants to study the function of the C-tail. So to put this in the structure here, when I say del7, it means it's deleted till here, del 13 would be somewhere here, and del 17 is all the way up here, the whole tail chopped off. So this is where we began the process, and we kind of compared and contrasted the function of the C-tail with these tail deleted mutants. First observation that we had was rather serendipitous because we were not expecting it. So we wanted to see the role of C-tail in DNA binding. So we use a classic biophysical technique, fluorescence and isotropy, where we attach a fluorescent probe at the end of the DNA. And when free in solution, this is free to tumble around in all directions. But however, when it binds to any protein, its movements are more restricted, and you see, and this is measured in terms of anisotropy, increase in anisotropy. What we found, so on the x-axis here, you see I'm trading with the protein, and I'm monitoring fluorescence anisotropy on the y-axis. What we found is for the MTF wild type, and even till if you delete 12 amino acids, there is no binding of MTF to DNA. However, on deleting 20 amino acid, there was clear binding of the protein to DNA, indicating that if you have the tail, the protein cannot bind to DNA. But if you delete the tail, now the protein acquires DNA binding properties. From this, we concluded that the C-tail kind of has an autoinhibitory function where it prevents the protein from binding to the DNA. And we tested this property across substrates. So here you can see the function, here you can see the KD or thermodynamic binding constant across substrates. And we found that there was no preference for any DNA substrate, which means that it indiscriminately binds to any DNA that is given. So from here, our main conclusion was the C-tail is autoinhibitory when it comes to DNA binding. Next, we wanted to test if C-tail has any role in promoting complex formation with mitochondrial polymerase. So for here, we used another classic biochemical technique, which is biolayer interferometry, which is here what we do is we have, so this is SPR kind of a technique where we have a biosensor which has a specific probe attached to it. In this case, we immobilize his tagged MTF. So this is a nickel probe, NINTA probe. So we immobilize his MTF and we tested the binding of the polymerase to the factor. When there is just MTF, no binding, the way the light is reflected will be different from when there is the polymer, the bigger protein bound to the surface, and the light will be diffracted differently. So what we found here again on the x-axis, I titrated with the polymerase at constant factor concentration, and on the y-axis, I monitored binding. We saw robust binding between the polymerase and factor when the C-tail was present. However, when on deletion of the C-tail, there was a very little binding between the polymerase and the factor. And this helped us conclude that it is really the tail that's helping in the interaction between the polymerase and the factor. So we concluded from here that the C-tail promotes the complex formation between the polymerase and the factor. Just to give you a summary of what I told you till now, so the factor really exists in two states: a free state where the tail moves away from the protein, and an autoinhibited state where the tail folds in on itself, that kind of closes its DNA binding, stops its DNA binding ability. These two states are in equilibrium. When the polymerase comes in, this inhibition is relieved, and now the factor can interact with this polymerase DNA complex with the help of this C-tail. So the C-tail here has two functions. One, it is maintaining an autoinhibited state. Second, once released, it helps in the interaction between the polymerase and the factor. The next thing we wanted to test is its role in transcription. So we first deleted the tail and we saw, is this active in transcription? So we ran what is a classic sequencing gel. So what this is, it is a basic polyacrylamide gel. It is a very high percent polyacrylamide gel. This is a 24% gel, and why that is important is that high percent gel helps us resolve up to single nucleotides. So what I did in vitro transcription reaction in a test tube with gamma ATP that will label the first nucleotide of my RNA. So essentially what you're seeing is labeled RNA on a high resolution transcription gel. So at the bottom is my free gamma ATP. So this is like the 1 mer. This is what was not used in the reaction. At the bottom is the free gamma ATP, followed by 2 mer RNA, 3 mer RNA, 4 mer RNA, up to runoff. So this gel helps me resolve all the small RNAs that are present during transcription initiation. As you can see, the stark observation here is, as you can see, on deleting the tail, there is a prominent decrease in the amount of runoff RNA or full-length product that is formed, which is quantitated in this figure. So we knew that if you delete the tail, yes, you're defective in transcription. So now that we know that the tail has a role in transcription, we came back to our original figure where we can dissect step by step where is this defect coming from. So we know the C-tail helps in transcription, but which step does it help in transcription? So for that, we took it step by step. So let's look at the first step. This is the formation of the enzyme and DNA to form the closed complex. We can monitor this by fluorescence and isotropy, and that is exactly what we did. We added, we have a labeled DNA duplex here, and we added RPO MTF complex, and we measured fluorescence and isotropy, and this gave us binding of the strength of the initiation complex. As you can see, on deleting the tail, there is up to almost a 7-fold decrease in KD of the initiation complex. As you know, KD or thermodynamic binding constant means that the higher the KD, lesser is the affinity of the complex. Lower the KD, tighter is your complex. So we found almost a 7-fold defect in formation of the initiation complex. But we think that under physiological conditions of the proteins, this should have very little effect on promoter binding under physiological conditions. Thus, the first step, at least, was not important in terms of the tail. The tail does not have a major contribution here. So then the next step, we wanted to monitor what's happening next. So once this complex is formed, the next step is forming the open complex, right? Wedging open the DNA. So we wanted to monitor this step. And in our lab, we have a very interesting assay to monitor this. We do this by the formation of 2-aminopurine. So what that is you know in your DNA, the normal A residue, the adenine residue is 6-aminopurine, meaning the carbon 6 here has this amino group. What we do for 2AP, this 6 residue simply is moved to the second carbon. And by simply this modification, we have fluorescent adenine. The residue now becomes fluorescent, and we don't really understand why that happens. However, this is a great tool for studying DNA replication and transcription. And how we do this is you can buy this from IDT. So what we do is we replace a naturally occurring adenine residue with a 2-aminopurine residue. And when in duplex, when the DNA is in duplex, the 2AP fluorescence would be quenched due to base stacking. However, when the proteins come in and the DNA is now wedged open, if your residue is in the melted part, this quenching is destroyed, and now the base flips out and the fluorescence is enhanced due to this unstacking of the base. So if you see increase in fluorescence, as the readout, it means that your base has now become unstacked, which means your base is now in the melted region. So using this assay, biochemists often study DNA repair and transcription mechanisms. So we use this mechanism to monitor closed to open complex or the promoter melting, as we commonly call it. So what we put is, so the extent of the melted bubble for these mitochondrial system is -4 to +2. It's a very small bubble that exists in this system. So I put fluorescence bases in the -4 and +2 positions, one at a time. And what I measured is that when you add, so you can see in the DNA duplex, there's a very baseline fluorescence. When you add the polymerase, it gives you a little fluorescence, meaning there's a little opening of the duplex. And on addition of MTF, there's almost a 40-fold increase in fluorescence, meaning that the factor is what helps to melt the DNA open. When I compared this melting, the extent of this melting to the C-tail deleted mutants, I did not find a significant difference from the wild type for the -4 or the +2 position. And this means that the C-tail does not have any significant contribution to promoter melting or formation of the open complex. So this step, we saw a minor effect. This step, we saw there is no effect. And now we bring in the NTPs. So the next step, once the DNA has melted, the next step is binding of the initiating nucleotides and the subsequent formation of the bond between them, that is a phosphodiester bond. And we can monitor these steps, the kinetics of these steps, using fluorescence and gel electrophoretic methods. So we had used the, just as a reminder, our lab had defined the extent of the melted bubble in the mitochondrial complex. And how we did this is simply by the 2AP method. So we put 2-aminopurine in place of pre-existing adenines. Naturally occurring adenines were one by one replaced by 2-aminopurine. And in this way, we found that the extent of the bubble was -4 to +2. Curiously, what we had found is that there is a ATP sensitivity for the -1 position, meaning that if you titrate with ATP at the -1 position, there is a subsequent increase in fluorescence. And using this as a signal, we were able to determine the KD, or thermodynamic binding constant, of ATP for the initiation complex. And we determined it to be around 90 micromolar. I wanted to exploit this property to see if the C-tail has any contribution to binding of the ATP to the complex. So what we are doing here in this assay is simply putting 2-aminopurine in place of the naturally occurring -1 adenine. And what we do is we monitor the flipping out of this base, and we monitor that. So I am titrating on the x-axis +1 and +2 initiating nucleotides, the first two bases that will come in to initiate transcription, and I'm monitoring fluorescence intensity in the y-axis, which will give me a binding curve or binding curve. So using this method, we are monitoring this, the open to initiation complex, and then binding of these NTPs that is shown with these orange blobs. What we found is that we saw a robust binding when the C-tail was present, and however, on deletion of the tail, there was the binding is definitely weakened when we delete the tail, which is, and this is quantitated here. As you can see, the KD for the initiating NTPs is much higher, which means that the affinity of the complex for the ATP is much lower on deletion of the tail. So from this, our conclusion was this C-tail deletion causes a severe defect in initiating nucleotide binding. The next step we wanted to monitor was the actual bond formation between the two nucleotides that were bound. So now that these nucleotides are bound, there should be some sort of bond formation, and we wanted to find out the kinetics of these bond formation and whether that is affected by the C-tail. We used a transcription-based assay, a kinetic assay, and we found, as shown in the graph, that there was a decrease in the rate of tumor synthesis on deletion of the tail. And this decrease was contributed both by Km, which is affinity of the nucleotides for the complex, and Kcat, which is the rate of the phosphodiester bond formation. And both of this together combined contributed to the decrease in the tumor synthesis rates of this complex. So from this, our conclusion was the MTF1 deletion causes a severe defect in initiating nucleotide binding and RNA priming. The last result that I want to discuss is, so all these assays, all these biochemistry that I've shown you are bulk assays, meaning you're measuring average behavior of molecules in your assay. We wanted to specifically study subpopulations and behavior of a single molecule and how that is affected due to deletion of the tail. So what we did is we did some single molecule fret time studies, and this helped us measure the dynamics of the open and initiation complexes of MTF wild type versus the C-tail deletion mutants. So let me explain this slide a little bit for those of you who are not very familiar with single molecule studies. So how we did it, so this was my DNA sequence, and basically you can buy this. We just attached Psi5, which is the red or pink probe, at the -16, and Psi3, which is the green probe, at the +16 position. And -16 +16 is, of course, in reference to the +1, which is the transcription start site. So now, what we did is we have a quart slide, and we immobilized this DNA, this piece of DNA, to this quart slide to be able to study it. When it is free, this DNA is kind of straight, and however, when the proteins come in, the transcription the factor in the polymerase bends this DNA, which causes these two probes to come closer to each other, giving you a fret signal. So using this kind of system in transcription, we often study the bending behavior of the DNA that gives us an idea about the dynamics of the closed, open, and initiation complexes. So this was our system. Simply, there are two probes. If the DNA is bent, they come closer and you see a signal. If they are far away, fret decreases, you see lesser of a signal, and you know your DNA is unbent. So let me explain a little bit how we read these single molecule data. So this is the Gaussian distribution for my molecules. So by position 0, I mean that there is no NTP that has come in. So it's just the DNA bound to my factor and the polymerase. So when you have only DNA, you have, as you can see, a very low fret value, meaning these two probes are far apart from each other, and the DNA is almost straight. And thus you see a very little fret value. Once you add the proteins, which is your position 0, because there is no nucleotide yet, once you add your proteins, you can see there is a second population shown in red that appears, which is at a higher fret value, meaning that your DNA is now a little bent because the probes have now come a little closer to each other, which causes this increase in fret value. So in addition of the proteins, there we see an appearance of a new population shown in red, and this red population is a higher fret value. So now there are two populations in your system. One is the free DNA, which is unbent, which is almost straight. The probes are far apart, and E fret value is low. Second, the DNA is a little bent because your probes are now closer to each other. The E fret value is a little higher. When you add first nucleotide, which is in position 1, there isn't much difference as compared to position 0. However, when you add the second nucleotide, which is the +2 position, which is in this case is GTP, there appears a third population or molecules that are in higher fret value, almost 0.6. And this means that the probes are much closer to each other. The DNA is more bent, and this means that the probes are very close to each other, higher fret value, and this is your third population, which is shown in blue. Thus, for position 0, there was an equilibrium between two fret values shown between these green and red populations. In addition of the nucleotide, the majority of the molecules DNA molecules are now in an extremely bent state shown by this blue population, and there's a decrease in population for the red and green population. So this is how we read a Gaussian curve. And then here you can see the individual time traces. So the green lines indicate the Psi3 probe and the pink lines indicate the Psi5 probe. And the blue lines is kind of these two combined readings. So as you can see, for position 0, there is a equilibrium between two fret states, which is shown by this blue curve. As you can see, they go between these two states mainly. This is the same for the +1 position, but the +2 positions, the higher fret state is dominant. And you can see that the time traces are mainly in the higher fret values, and they very rarely go to the lower fret values. So the majority of the molecules are in this higher 0.6 fret state. So this was our findings from the wild type, and now we deleted the tail and put it on the slide to see what happens now. So basically, you have seen the left panel. I explained it in the last figure. So for position 0, for when you have the tail, we saw that there was an equilibrium between a green state, which we call the closed state. The DNA is now almost straight, to a red state, which is like which we annotate with this red peak annotates kind of the open state, where the DNA is now slightly bent and open. However, for the position of 0, when you delete the tail, we saw a wider distribution of this open state, and we saw that it is not also distinctly differentiable. You can see the peaks here are pretty distinct, but here there is distinct overlap between the closed and the open state. And this means that the molecules are traveling between these two states more freely than when you have the tail. This does not change much in the +1 position, but in the +2 position, we can very accurately see the difference. So if you have the tail, most of the population in this state with 2 nucleotides shifts to the bent or the severely bent or the bent state, severely bent state, which is shown by the blue population. However, if you delete the tail, this blue population is not that distinct, and there is overlap between the green, red, and the blue populations, meaning that the molecules are sampling all the populations. They are kind of distributed between all these fret states, meaning there is more distribution of this molecule. There is no distinct one population. And you can easily see that with these time traces. So for the position 0, the MTF is mainly sampling between the open and closed state, but on deleting the tail, it is mostly in this open state, the higher fret state shown here by the red diagram, by the red population of molecules. Similarly, for the +2 positions, for if you have the tail, the molecules are mostly in the higher fret state, which is shown here by this population. They're mostly in the higher state, but if you delete the tail, they sample all populations, which is shown by the fluctuations, extreme fluctuations in this tail deleted mutant. So from this, our conclusion was that the tail kind of helps bend the DNA and kind of keep the DNA in a particular conformation, which is not happening if you delete the tail. Now the DNA is fluctuating and moving in all possible conformations. We also measure the kinetics or the transition rates between these closed, open, and scrunched complexes. And as you can see, on deleting the tail, the rate of a transition between the closed and the open state is much increased as compared to when you have the tail. Similarly, the between open and scrunched state, the rate of transition is also significantly affected if you delete the tail. At the end of my talk, to put it all together, so what does the C-tail of mitochondrial transcription factors do? Firstly, they are autoinhibitory. They keep the factors in an autoinhibited state. And if they move away, now the factor is free to interact with the polymerase through the interactions with the C-tail. So the C-tail is an on-off switch. If it binds on itself, transcription is off. If it opens, now it can go bind to the polymerase. So now this is in the on state. So the first function is a C-tail is an on-off switch for mitochondrial transcription. The C-tail is kind of turning on or turning off your transcription in your cell. Second function is helps in formation of the initiation complex with the polymerase. There are interactions of this C-tail with the polymerase that helps form the closed complex. So it is facilitating complex formation with the polymerase. And after this complex formation, the DNA is melted open. The C-tail does not have a role here. This green strand, which is your template strand, is aligned deep into the active side, which is shown by the dark gray sphere here. And now in the next step, the initiating nucleotides will come and bind to the template. And here the C-tail has a crucial function. The C-tail is pulling the template deep into the active site. It is like a placeholder. It holds the template in the active site in a stable conformation so that which facilitates binding of the initiating nucleotides to the initiation complex. If you don't have the tail, the template is wobbling around trying to find a stable conformation, and that's why we see sampling of the population in our single molecule studies. But if you have the tail, which is the placeholder, it pulls and keeps the template strand deep into the active site that helps binding of the initiating nucleotides. And this makes the process of transcription very efficient. So the tail is increasing the efficiency of your mitochondrial transcription happening in yeast and human cells. Finally, we have formation of the 3-mer, 7-mer, 8-mer, and we think that the tail is ejected at this step, which transitions the complex into elongation. So overall functions of the tail is autoinhibition, formation of the initiation complex, and acting as a placeholder that facilitates mitochondrial transcription. I would like to thank my advisor, my committee members, my lab members, and our funding sources from the NIH and the American Heart Association for funding my research during graduate school. Thank you for coming to my talk, and I'll be happy to take questions.
Awesome. That was really in depth. I'll tell you what, let me go ahead and keep the PowerPoint up as well.
Okay.
So that way, you can go back and forth through it. Okay, so we'll just go ahead and jump straight into the questions. So the first one here, and so some of these you may have touched on a little bit, after the questions were asked, but I'll kind of go through them anyway. How does the composition of the C-tail affect its activity?
Yeah, so we have not individually looked at the acidity or basicity of the amino acids and dissected that out. But we do have a couple of acidic residues in the tail, which we thought would help it act like a DNA mimic. It would give like an overall negative charge and it would make it act like a DNA mimic. And it would be, that is what we think is causing the autoinhibition. Like there's a DNA binding channel which is positively charged, so the negative DNA can bind to it. The tail itself having negative residues is kind of mimicking the DNA and closing off that DNA binding groove. So it's like a DNA mimic, but we haven't individually dissected the amino acids.
Okay, that makes sense. It certainly seems like it would be something difficult to do. Okay, so you talked a lot about the different factors that come in to help the binding and then I guess the transcription. Is this something that we could create a synthetic version of? Like could we make or could we mimic the factors to help drive the transcription if that's something that we wanted to do?
That's a very interesting question. I don't know that. Yes, I guess that would be an interesting thing to do. But I guess so. We're talking about making a synthetic protein that could act as factors.
I think so. Yeah.
Yeah, I actually haven't read any reports of that, but that's a really interesting question.
Okay. Maybe more generally, is that something that we would even want to do? Like, are there situations where I don't know. I'm way out of my depth on this one.
Yeah,
Is
I
there?
think mitochondrial transcription
Mhm.
is crucial in cancer cells
Okay.
because sometimes we see that mitochondrial transcription is turned up.
Mm-hmm.
So if you have a handle on this on-off switch, I think that would be a very interesting way of controlling the proliferation of cells.
Okay.
So that would be really interesting.
Okay. Interesting. Let's see. So you touched on, I think, well, you sort of touched on this a little bit, but can you track the time that it takes transcription to happen with transient fluorescent measurements?
Yeah, so those are steady state measurements. So you can actually track in, I think, up to milliseconds, as far as I know,
Okay.
to as to how this transcription is happening.
Okay. Wow, that's very fast. Do you have to worry about like the background fluorescence at all, or are you able to control it pretty well based on the binding that occurs?
Yeah, so for the background fluorescence, you have to have an absolutely duplex DNA to control the background fluorescence
Hmm.
because anytime the probe is in single strand, floating around in single strand, you will have some fluorescence. But if you can ensure that, so what we do is when we anneal the DNA,
Hmm.
one strand is labeled and one strand is unlabeled.
Hmm.
So we always add excess of the unlabeled strand so that
Okay.
all the fluorescent strand is in duplex and the background is kind of lowered.
Okay, that makes sense. So it could be an issue, but you basically take steps to try and
Yeah,
minimize. Okay,
yeah.
got it. All right. Okay. So this was a good question. In the part where you were talking about attaching the um, fret probes, how do you control where the probes actually attach? Like you listed. This was a question I had too. You listed specific places or specific spots on the DNA strand where the probes got attached. How are you able to control that?
We get it ordered from IDT. IDT will do it for you. They will exactly attach the probe where
Ah,
you want to.
okay. Well, that's really handy. That's really nice. Okay. And in that same area, I got a question. Does that broad distribution of the state of the complex in the deleted tail molecule mean that it doesn't transcribe as readily?
Yes, yes, that absolutely means that it will not transcribe as efficiently as the wild type. And I think I showed that with one of the old, with one of my, what happened here? With one of my transcription gels.
Mm-hmm.
So in the first slide, if you remember, the runoff is severely defective as compared to the wild type if you delete the tail.
Oh,
So
okay.
so the transcription is absolutely affected. It's not an efficient process anymore because
Mm-hmm.
the DNA is simply kind of sampling all those. It's kind of wobbling around, like trying to find the stable
Right.
information, but it's not an efficient system. And that's why in the readout, the RNA synthesis suffers.
Okay. So basically, the tail takes it from like a random walk type of thing to a very controlled, or at least a more controlled
Yes,
uh,
it takes it into a
process.
very controlled and stable conformation.
Mm-hmm.
So now it's like bam, bam, transcription can happen super efficiently.
Okay, got it, got it. Okay. What another question I had was sort of about how you determine the significance of something. So, and I don't remember exactly which slide it was on, but you were showing that the presence or absence of the C-tail didn't have an effect on a particular part of the step. I think it was the open complex formation maybe.
Uh-huh.
And you had some bar charts that showed basically this one right here. That's it exactly. So how do
Yeah,
you
yeah. So
what's the
we can.
cutoff there? Because like I see it goes from like 40 to 60 fold change. At least
Mhm,
roughly on average. So
mhm.
how do you know that that's not a significant difference?
Yeah, so the thing with these fluorescence assays is it can vary between days of doing the same experiment
Mm-hmm.
because remember there are lamp fluctuations,
Oh yeah.
etc. And so every day you turn on the machine, you will not get the same data.
Yeah.
If you get the same data, that's something to worry about actually.
Sure.
So there is a lot of fluctuation. So we kind of do so this has to be done on different days, and even done like in my hands it will vary quite a bit.
Mm-hmm.
So we always take kind of an average. I think this is average for 3 or 4 different readings from different days done on different days, and then we kind of average it out, and we look at the error, and then we kind of measure the difference there.
Mm-hmm.
And this was not significant enough for us to claim a change.
Okay.
So
So if the delta 20 was at like, let's say, an 80-fold change, then you might step
yes,
back and say,
2 to 3. If it was 2 to 3 fold, we would look at it, but this is not really
okay,
that.
yeah, that makes sense. I know that's sort of a rule of thumb that I've heard in some of the work that I've done, like in electrochemistry. I think a really simple one is like if signal to noise is if the signal to noise ratio is like 2 or 3, like you said, then you start looking at it and going, okay, is it
Yeah,
is it really a significant a significant change, or is it just what it kind of happens to look like?
yeah, yeah.
That makes sense. That makes sense. It's really fascinating work. I especially like the way that you sort of step through each part of it and said, okay, here the tail's not doing something, here the tail does a little bit of something but not much. Here's where it has the really big effect. And it's fascinating to see. Like, so this is way outside of my field, but you've presented it in a really you've taken something complex and made it easy to understand. At least for some
Thank you.
not I don't think I could do this work, but I understand what you're doing, and that's really cool.
Thank you.
So I guess I'll ask one final question because I think that I've gone through all the other ones. Is this technique something that could be applied more broadly? Like, could you take some other system? Again, I don't really know enough about it to pick one, but like, could you apply the steps that you've gone through here to other systems to understand, what part of a complex is driving which part of the, let's say transcription or something like that?
Absolutely. I think the techniques used here, especially we have used uh, protein DNA binding assays, like I showed here. This is like classic fluorescence anisotropy that is used widely in a lot of biochemistry applications.
Okay.
So this is definitely has a huge broader implication. And then we have used the Blitz system, which I showed in the beginning, which is here. This is a standard SPR kind of a method that is used for a lot of wide applications, like looking at inhibitors binding to proteins, etc. And so this is a very, very common method. I think in industry they also do this high throughput. And so I think a lot of these techniques are very like classic fundamental biochemistry method, and you can definitely use these techniques as general to study these processes.
Okay. That's really cool. Like I said, it's really neat to see the way that you're doing all this work to really get at some fundamental questions. And it's just really cool to see how it all comes together. So it looks like that's all the questions that we've got. So we'll go ahead and start wrapping up. If I could get you to hang on for just 1 minute, we'll talk very briefly. But thank you to everybody for coming out. Appreciate the questions, and we will be back next week. We have Dr. Danny Arias Rotondo from Kalamazoo College. So I hope I will see you all out here next week. And in the meantime, have a great weekend. Stay safe and get vaccinated. And we'll see you all next week. Thanks a lot.
Thank you for coming, everyone.